Hey everybody, welcome to the first week of Microbiology 2. In this week, we are going to be covering mycology. So I am going to share my screen so that we can go through the PowerPoint. Again, this PowerPoint is posted in your doc sharing, so you should all have access to it there for you to go along with me. All right, so let me just put it into slideshow format, and we will get started. All right, so we were just going to go through the objectives here. You can read through them on your own if you'd like. But essentially, jumping into mycology, general characteristics of fungus. Fungus are eukaryotes, meaning they have a nucleus, they have mitochondria, they have thick cell walls made of chitin. And there's basically two groups of fungi in the lab, in the clinical lab. It's yeast and molds. So yeast tend to look very similar to like bacteria growing on an agar plate. They can be moist, creamy colonies. So yeast, and sometimes they can even be dry. They're not always truly that mucoid looking. Sometimes they're very dry looking, but they grow on agar similar to bacteria, and we can grow yeast on regular blood agar. So just kind of keep that in mind, whereas a mold is going to be the classic fungus that most people think of. When you think of molds growing on your food, this is what you're going to get. You're going to get that different textures, like fluffy, cottony, woolly textures. And depending on the type of mold it is, the different type of texture, the different color, that kind of thing. Just so here is a yeast. So again, you can see how similar it looks to bacterial colonies growing on agar plates. And then again, molds definitely have that texture to them, that powdery, cottony, whatever you might want to call it. All right, some basic terms to understand just a little bit more on the molds. Hyphae, we do refer to hyphae when we look at molds under the microscope. And so hyphae is the basic structural unit that make up a mold. And it can be two different types. You can have septate hyphae or aseptate hyphae. And septate hyphae are going to have the cross walls, whereas aseptate hyphae have none. And so there is a category of aseptate hyphae molds that are the zygomycetes. These category molds are very well known to be aseptate. Otherwise, the majority of molds fall into the septate. Most have the cross walls. And then if you ever hear the term mycelium, that is referring to all the hyphae together. And the hyphae is what allows for penetration of nutrients to help keep that mold growing. So here is showing the mycelium with all the different hyphae. Hyphae can branch up into the air, what we call aerial. They could go down into, like in this case, the agar plate to get nutrients, vegetative. Here is what those cross walls look like for septate hyphae. The arrows are pointing to the cross wall there. So that is what septate hyphae would look like, whereas here you don't see any of those cross walls, so that's aseptate. There are different shapes that your hyphae can take on, from antlered, racket, spiral, rhizoids. The only one that I am concerned with for us is rhizoids. So this last picture here, rhizoids tend to have what we call a root-like structure. So they kind of look like roots under the ground. In this case, in the picture below, it's showing a real-life picture of rhizoids. So that is key. There's 3 organisms that we will discuss that are differentiated based on if they have rhizoids and where the rhizoids is at. So just keep in mind, the only hyphae shape I want you to remember is the rhizoids, the roots, basically. And then we will cover those 3 organisms that are dependent upon knowing where their rhizoid location is. All right, going a little bit into reproduction. A lot of times there's reproduction asexually through conidia or arthroconidia. So there's different types of conidia. I am not going to test you on all this. Instead, we're focusing clinical microbiology. So we're going to focus more on the main molds and yeast that we see with diseases and infections in the lab. So this is background information. It's here. It's in your book, well explained. So if you want to read through how they reproduce, you can do that. But I will say at the very bottom here, the number one way that we identify molds is by microscopic identification, viewing them under the microscope. And when we view them under the microscope, a lot of times we're looking at the conidia, which is the asexual reproduction part. So it's just kind of a side note there. All right, so here's just some pictures of how those little conidia look. They look like little spores that you breathe in. Here is our conidia down at the bottom. Again, none of this is going to be tested. It's just giving you some representation of it. All right, so identification molds, growth rate. Molds take a long time to grow. This is not a fast process. There's nothing stat about identifying molds. It takes forever. If you think about it, a lot of times, how long does your food have to sit on a counter before it finally grows mold? It's not going to be right away unless your food is already old when you bought it. It's going to take a couple weeks, for it to get something. So it's the same in the lab. It takes a long time for us to grow anything out. Once we do grow it out, we do take in the colony morphology, just like we did with bacteria. We're looking at the color, we're looking at the size, we're looking at in this case with molds, especially texture. So all of that is written down and taken into account. And again, the best way to diagnose and identify is through the microscope. We need to see what it looks like under the microscope, and that is how we determine what it is. So here's the categories of fungi. We have zygomycetes, ascomycetes, basidiomycetes, and deuteromycetes. So in the zygomycetes category, these are the ones that are the aseptate hyphae, the no cross walls. And here are the three at the very bottom. We have Rhizopus, Mucor, Absidia. And I do want you to know those three. These are the three that I'm going to reference those rhizoids in. Remember, the rhizoids were the root-like structure. These are the three that are dependent on where that root-like structure occurs on how to differentiate them. So this is just showing, oh, by the way, they reproduce through sporangiospores. So they make spores in these like fruit structures. And so they're just showing you this here. Here's a real life picture. Aren't they pretty? Molds are so pretty under the microscope. I love them. They're not pretty on your food, but they're pretty under the microscope. All right, so again, this is just background information on how they reproduce and make these spores. But the key, what I'm getting to, is the rhizoids, the contact points with root-like structures. So again, these three are going to be based on if they have rhizoids and where they have them. Overall, the infection of zygomycetes, it infects mostly immunocompromised. You're going to find that with the majority of fungus, it's usually infecting immunocompromised patients. It is amazing how much we are breathing in mold every day in our lives. We are exposed to fungus on a regular basis in the outside environment everywhere. But most of us have a really good immune system that it doesn't cause us a problem. It's the immunocompromised patients that really end up getting this. And so in this case, zygomycetes really tend to affect people that have poorly regulated diabetes. If their diabetes is really out of control and their immune system is kind of shot, they might pick this up. You'll hear a lot about like AIDS patients picking things up. Anybody that has just bad respiratory systems, they'll pick up fungal pneumonias, things like that. So just keep that in mind. It's typically immunocompromised. And again, we're typically getting them by inhaling the spores. So in this case, zygomycetes, again, infects immunocompromised. It can be found throughout the environment, inhale the spores, and it can become quite progressive. It will go not only just to the lungs when you inhale it, but into your GI tract and so on. So here's our first one out of the zygomycetes, the Rhizopus. And letter A is just showing you the sporangiospore up at the top. B is their hyphae, and C is those rhizoids. Do you see how it's those root-like structures there? So the key with Rhizopus is we know that the rhizoids always occur at the base of that hyphae. So you can see it's at the very bottom of that hyphae there, and that is the key on Rhizopus, remembering that the rhizoids are at the base. Absidia, so letter A is the sporangiophore. There's actually two of them. They only labeled one. And then you have two hyphae going down, and this is a tricky picture, I know, but B is showing the rhizoids here. So here's that root-like structures again, and it's actually occurring between the two hyphae here. It's hard to see in this one, but there is two hyphae, and it's occurring right between them. So that's always key, is rhizoids between for Absidia. And then Mucor has no rhizoids. So there is no root-like structures. It's just the sporangiophore with the simple hyphae down, if you will. So no rhizoids here to worry about. So again, know those three. Those are all three aseptate hyphae, and knowing if they have rhizoids and where they occur. All right, into our next category is ascomycetes. The important fungi that occur in this category is Microsporum, Trichophyton, and oh, this is a beautiful word. Oh my gosh, Pseudolascaria boydii. Oh my goodness. You get the idea. Again, these do have septates because the rest of the categories will have septate hyphae. Again, reproduce asexually by canadia, reproduce sexually by something we call ascospores, thus the name for the category ascomycetes. Basidiomycota is also forms basidiospores, that's why they named the category. The only clinically important one here is Cryptococcus neoformans, so we'll get to learning about that in a minute. And then our last one, Deuteromycetes, basically involve the biggest number of infections that we find. They involve everything from cutaneous infections to subcutaneous to systemic. So most of the mycology, most of the yeast and molds that we're going to talk about fit into this category. So that's just kind of a quick overview on the other three categories. So we are going to get into, let's discuss the actual clinically significant ones here. So we are going to go by disease state. So we're going to start with learning all the fungus that cause cutaneous infections. Then we'll go into subcutaneous, systemic, and opportunistic. All right, so cutaneous. There's a lot of fungal infections that involve your hair, skin, and nails, all of which we consider cutaneous, or what you can call the dermatophytes, derma for skin. So they don't typically invade deeper than that superficial level. They usually just keep it. But you're going to hear words such as tinea, tinea nigra, piedra. These are the terms that we define as these cutaneous infections. So here it's just showing you a representation of the skin layers. So it will not go anything below the dermis layer here. It's going to stay at the dermis layer and above. All right, so the main three categories that are known to involve infections of the hair, skin, and nails are Trichophyton, Microsporum, and Epidermophyton. So, and then here is all the different types of infections that you might hear. And the second word is going to tell you where on the body that infection is occurring for on the skin, hair, or nails. So, for example, tinea capitis is an infection on the head, cap for head. Tinea favosa is a distinctive part of the head. Don't worry about that. Tinea barbae is beard. Corporus is the body. Menuum, hand. Inguium, I can never say it right, nails. Crurus, groin. Pedis, feet. So you can kind of get the idea. Anytime you hear one of these words, it's just referring to where that skin infection is. All right. I did not mention this on this slide here, but it is important for you to remember Malassezia furfur. Malassezia furfur is a yeast, and it definitely involves the skin. That's why it's still part of this. It's actually lesions on the chest, trunk, and abdomen. And basically, this yeast has a very unique, what we call spaghetti and meatball appearance under the microscope. It's very well known for, again, immunocompromised patients. So please make sure you remember Malassezia furfur. Remember, it's a yeast, and that it has this spaghetti and meatball appearance. And here's that spaghetti and meatball appearance. So, I mean, just use your imagination. So I do want you to be able to identify this picture. So on your quiz and upcoming exams, there will be pictures because number one way of identifying molds and yeast is through microscopic. So you do need to be able to recognize Malassezia furfur as the spaghetti and meatball look. We will practice some of these pictures in the lab on Thursday together. All right, Piedra horte is what we call black piedra. This forms hard black nodules around the scalp hair, and you can see that here in the picture. So that's why it's part of the dermatophyte family. You just need to remember that Piedra horte is the cause of black piedra. There's not a microscopic picture that I'm going to make you remember on this. In this case, just remember it does the black nodules around the scalp hair. Trichosporum is a white piedra on the hair shaft. It does look like it's basically a yeast, so it'll have a yeast-like appearance. It'll have a yeast appearance on agar. It'll be creamy, smooth. Here it is under the microscope, so you can see the yeast look. Yeast are tend to be ovally in shape. If you see them budding, it's a lot easier to tell. I am not going to make you remember the look of Trichosporum. It's just a mention of it causing white piedra. Horte wernickeyi causes tinea nigra, which is a brown to black look to the palms and feet. And you're not going to have to identify this. You just have to remember Horte wernickeyi as causing tinea nigra. All right, one note though, you'll notice here you saw skin scraping and KOH. Anytime that we have a yeast or fungal infection in general suspected, a lot of times they do what we call a KOH preparation of potassium hydroxide. And usually it's with a skin scraping. So anytime somebody comes in and looks like maybe they have ringworm, which ringworm is caused by a fungus, just so you know. I know it says worm, but it is a fungal infection. Or any other type of fungal infection that look to it, they'll do a scraping and send it to the lab. And what we do is we'll look at it under the microscope with nothing on it really at first to see if we can see any hyphae or yeast. And then we'll add KOH, potassium hydroxide, to it. And what that will do is help dissolve any skin cells. So any of those epithelial cells, all that stuff that might cloud up your view, that will dissolve it all. And that way, if there is yeast or hyphae present, it'll come through and we can see it easier. And so it's a simple reporting. You look at it, you look for yeast or hyphae. If there is yeast or hyphae there, you just say present. If not, nothing, and that's as simple as that. And it gives a quick view to the doctor of, yep, this is a little bit of a fungal infection. So that's all you have to do. So that is a very common thing done in the lab, is the KOH prep. All right, continuing on. Trichophyton, a dermatophyte, again, involving the hair, skin, and nails. In this case, it tends to have what we call macroconidia, which are larger conidia with 3 to 8 septates. There are several types of trichophyton, so we're going to go through those. Trichophyton rubrum, when it grows, it is a mold, and when it grows out, it will be white to red, cottony, velvety on the top. And then the neat thing is, when you flip that agar plate over that you grew it out on, the reverse side is a very classic cherry red color. It is so big for Trichophyton rubrum. It's a very classic look to it. So I bolded that there because I do want you to always keep that in mind. When you look at it under the microscope, you will see that it has cigar-shaped macroconidia and tear-shaped microconidia that are said to be birds on a fence look. So I will come back to the slide. I know I skipped some stuff. So here's what it looks like on the agar, the white kind of cottony on the top, and then that reverse side being cherry red. And then here's the birds on a fence with the microconidia. Isn't that little? It's kind of cute. You can kind of picture that they look like little birds. All right, going back to what I skipped. This is a very common infection to see in the lab with skin and nails, especially so tinea corporis in general. So please remember Trichophyton rubrum. All right, Trichophyton mentocrophytes causes more scalp lesions, tinea pedis for feet. So white, cottony, or yellow on the surface. You can see that here. The reverse side is more of a rose brown. The key with this one is grape-like clusters of microconidia, and then it also has cigar-shaped macroconidia. So letter A is showing you the grape-like clusters. Letter B is showing you the cigar-shaped look. So this is Trichophyton mentocrophytes. I am not going to test you on the picture of this, but I do want you to always keep in mind the classic grape-like clusters is known for Trichophyton mentocrophytes, but I'm not going to test you on the actual picture itself. So just keep that in mind. And then there's just another picture. So I'm not going to worry about these. I want you just to remember these characteristics overall. That's it. All right, Trichophyton tonsurans causes tinea capitis, so it's a head fungal infection. In this case, when it grows, it has these what we call radial folds and a depression in the center. So you can see that here, that depression in the center, and then these radial striations kind of coming out. So that's a very unique look to Trichophyton tonsurans. We're not going to worry about the microscopic view as much here. So just know the radial folds crater-like depression. Otherwise, you don't need to remember a lot here. Trichophyton varicosum causes lesions in different areas. It's a huge cause of ringworm in cattle. I think I have a picture. Yeah, there it is. That poor little cow. And then there you can see what it looks like on the agar plate, what we call button-shaped white raised center. I know you can't really see the raised center, but under the microscope, it has chains of chlamydospores, but again, we're not going to remember this. So in this case, there's not much I want you to remember on trichophyton verrucosum. It's just mentioned. There's so many organisms, but it's hard to remember them all. So don't worry too much about this one. But microsporum, we're definitely going to have a couple here that you will have to take away. So in general, microsporum are also dermatophytes. There's a note here, microsporum, that word, Odoonii, causes tinea capitis, especially in children. We're going to get to a couple other microsporum. In general, they have these big, large macroconidia that will have a very specific shape to them, and that's going to be what's going to be key in identifying them. So we're going to start with microsporum canis. It's pretty much mostly in animals, but it is the #1 cause of ringworm in dogs and cats, and thus the canis for the dogs. Humans can get this from contact with their infected animals, and they do. So I definitely need you to know microsporum canis. And the big key part is what's bolded at the bottom here. Under the microscope, it has these spindle-shaped macroconidia with 4 to 8 septa and curved ends. You have to know this picture. So you see how it kind of is that pointy ends there, the spindle shape, and then it has the 4 to 8 cross-wall septate there. That is key to identifying microsporum canis. There it is again. It's just not stained in this one, but you can see it. It's the same shape. And then the other microsporum I definitely want you to remember is microsporum gypsum. It doesn't cause much infection, but I do want you to remember because I want you to separate it from the microsporum canis. So with microsporum gypsum, in this case, the ends are more rounded. So it's not as like pointy on the ends, it's more of a rounded end. So I do want you to be able to differentiate those two. So there it is again. All right. Epidermophyton, there's only one member. It's Epidermophyton flocosum, and it causes tinea cruris and tinea pedis. It is a club-shaped macroconidia. A lot of times people call it beaver tails. You do need to remember Epidermophyton flocosum, and there is the club shape. So please know this image as well. All right, so that was a lot of cutaneous stuff. I mean, it's such a big category. So before we get any further, out of the dermatophytes, I believe I pointed everything out to you that you need to know. I know this gets confusing because there's so much info. Know the epidermophyton, know microsporum gypsum and canis. Out of the trichophytons, the only image I wanted you to remember was Trichophyton rubrum with the birds on a fence. The others, you can just remember, if I had anything bold, you can remember it, but you don't have to recognize images. Again, the Trichosporum, the Malaysia furfur, you did have to know, spaghetti and meatballs. The Piedra horte, the Trichosporum, the Hortea, you just had to remember the key infection that went with it. You did not have to remember pictures. So hopefully that helps define the dermatophyte category a bit down. You don't have to remember everything. It's a lot of stuff that can fit into that category. All right, going into the subcutaneous infection where we're getting lower than that dermis layer. Now we're under, and the part with this is there is one very large organism, not large, it's not like large itself, but one significant organism that I definitely need you to link with subcutaneous, and it's at the very bottom here, called Sporothrix shankiae. And it is a subcutaneous infection that will become or can become systemic. So Sporothrix actually fits into two categories. It fits into the subcutaneous category, and it will fit into the systemic category, which is one of the next ones that we have to discuss. So here we are for where we're at in the skin layers, is in the subcutaneous section. All right, so Sporothrix is the only one I'm going to have you remember out of the subcutaneous. It is living in vegetation, and it's what we call rose gardener's disease. So a lot of times people pick up this subcutaneous infection when they're gardening, and they might have that vegetation, say it's on roses, and the thorns poke their skin, and it gets in there that way, or splinters, or whatever it might be from gardening. And that's how people acquire Sporothrix shankiae. Now, Sporothrix shankiae is unique in that it actually occurs both as a yeast and as a mold. And there's going to be a few others that we'll talk about coming up. that do that as well. But there's only 5 that we need to remember in class that do that, and Sporothrix is one of them. So it'll occur as a yeast and it'll occur as a mold. Under the microscope, the key to identifying Sporothrix is it has what we call single-celled conidia that cluster at the tips of the hyphae. And so letter B is your picture for that. I don't care about the yeast look. I care about the mold look here, letter B. So you can see how there's single conidia or single hyphae that have these clustering at the tips. To me, they kind of look like little flowers almost. That is, you have to know how to identify Sporothrix. So that is essential to remember. Here it is again, the arrows pointing out the clustering at the tip of that hyphae. There it shows you more of that flower look that I think it has. But yes, please remember Sporothrix. All right, opportunistic, there are 4 basic ones that fit. There might be another one, I just haven't listed it here, but Aspergillus is a big one in opportunistic, Fusarium, Geotrichum, and Penicillium. So before I move into it, what it means to be an opportunistic is these don't always cause infection. Like you and I have probably been exposed to this a million times, but if you're immunocompromised enough, then it will cause an infection. But overall, they're not always related to diseases. But if the opportunity is right, that's why they call it opportunistic, then they will cause the infection. All right, so starting with Aspergillus, found hugely outside the environment, just like a lot of these others. You inhale the conidia, that's how you get the infection. If you are immunocompromised and pick it up, it has got a high mortality rate, unfortunately. The first Aspergillus that we know here is Aspergillus fumigatus. On the colony, it's white to blue-green. Under the microscope, it's said to have what we call a foot cell at the base of it. It looks like a foot, is why they call it a foot cell. But I just think it's not showing you the foot cell in this picture, so don't look for it. But isn't that pretty? It's just so pretty. But yes, you have to know the way Aspergillus fumigatus looks. Please know this picture. A lot of times, Aspergillus fumigatus is nicknamed farmer's lung because it's so heavily found in the soil, and then farmers like tend their fields and they stir up the dirt and the spores, and if they're immunocompromised, they pick it up, and that's why it's called sometimes nicknamed farmer's lung. All right, Aspergillus flavus has more of a spherical vessel, and you can see that here. It kind of looks like sunflowers, doesn't it? And then Aspergillus niger has a very large spherical vessel, and the entire thing is going to be covered in spores. That one you do need to know. So out of the Aspergillus, please know the fumigatus and the niger for sure. And by the way, it's named niger because on the colony morphology, it has a black dotted surface and will eventually become jet black. So just to keep that in mind. All right, Fusarium, definitely need to know Fusarium as well. They have very unique little sickle or boat-shaped macroconidia. So they kind of look like, if you want to call them canoe boats, sickle cells, like a crescent moon shape. But that is a unique look for Fusarium. So you should be able to easily identify this. There it is. It kind of looks like a bunch of bananas with that staining. All right. Geotrichum is basically causes a pulmonary disease, just like a lot of these, but it's kind of got that square rectangular arthroconidia look to it. And they kind of chain together like that. So that is Geotrichum. There it's stained, so you can see that nice square rectangular chain together. And Penicillium. Penicillium is the mold that first discovered the antibiotic penicillin. This has a very unique, what we call brush-like, or you can call it a paint brush. Look to it under the microscope. That will be key to identifying it on the actual agar. It's kind of a green color there. But here is the paintbrush. Some people think it looks like wheat, so whatever you think, but please know Penicillium. There it is again. All right, and then kind of something that just doesn't fit all together with the rest is what we call dematiaceous fungi, and that means they are pigmented naturally. Most fungi, when we look at them without a stain, are hyaline, which means clear. You have to kind of stain them to pick up their features a bit better. In a lot of these pictures, we've seen the stain look. But if they are dematiaceous, they already have a pigment to them. And the one that I ask people to remember out of this is Alternaria. So Alternaria is a pigmented fungus. So you'll kind of see here, here's the pigment. This is not stained, whereas all the other fungi, when we looked at them not stained, they were kind of clear looking. So this is the pigmented showing. So it's kind of got this, what they call an acropetal shape to it. And I think, yep, there it says that. It's kind of what you call elliptical or ovoid or acropetal. So it kind of tapers down to the one end. So you do need to remember Alternaria. And as far as infections, it kind of causes a variety, but it can cause cutaneous as well. We just mentioned it separate because of the fact that it can be pigmented, and that's unique. All right, and finally, our last category are the big bad boys, the systemic infections. There are 5 of them that I want you to remember. There's more, of course, but these are the 5 that I am asking you to remember. So these 5 are well known at causing systemic, kind of more than just one area disease in your body. They can spread. So all 5 of these are what we call dimorphic fungi, and that's bolded at the bottom. And I will explain that here. I think I have a slide on it, but that means that they can occur both as a yeast and as a mold. So we already talked about sporothrix here at the one of the last ones listed. Remember, that caused a subcutaneous, but can also cause a systemic. And sporothrix, I told you, both was in a yeast and a mold phase. The other ones here are blastomyces dermatitis, coccidioides immitis. That one you guys are going to recognize when we get to it. Histoplasma capsulatum and Paracoccidioides braziliensis. So these 5 you need to remember cause systemic and are all dimorphic. So here's the definition of what a dimorphic fungus is. It means it could be a mold or yeast, and it's going to depend on the temperature. So a mold form is going to occur more at room temperature, 25 to 30 degrees. A yeast is going to occur more at body temperature, 35 to 37 degrees. So please keep that in mind with the temperatures in which each phase occurs. And they are all highly virulent. They're really good at causing infection, especially if it's a susceptible host. All right, so I think this came right out of your book, kind of what they look like under the microscope and under the colony morphology on all of them. You can read that later. I'm not going to read to you. So just information out of and then here's what they cause for infection. Again, the reason I said you will recognize coccidioides is this is the one that goes with our valley fever. I'm sure every one of you might have met somebody that has got valley fever. Definitely occurs in the Southwest. So here in Arizona, it's big. So if you work in labs, especially like here in Arizona, New Mexico, even southern Cali, you're probably going to run into, valley fever at some point with somebody. All right, so let's go through each one of these. We have blastomyces dermatitis. The disease that they name it is blastomycosis, definitely found in North America. As it showed here, it's more the Mississippi and Ohio River Valley area. It will start as the lung, can involve the skin, the bone, multiple organs. Again, it's a systemic. It goes throughout. So the 2 forms, yeast and mold. Out of the 2 forms, I want you to remember the yeast phase. And in the yeast, it has a very thick-walled yeast cells, which is unique. And I'm going to show you, that's not the best look. Here, this is a better look. You can see how thick those walls are on the yeast here. So that is unique. And I do want you to remember this picture, this image on how to identify blastomyces in the yeast phase. As far as the mold phase goes, they said to have lollipop-like look. I'm not going to test you on that image. I'm going to test you more on the yeast image. But there's the mold image if you were curious. And again, all right, Coccidioides immitis, again, that's our valley fever, desert portion of U.S., Mexico, and South America. Starts in the respiratory, spreads throughout. Again, occurs both as a yeast and mold. I want you to remember the mold phase on Coccidioides. It will be a rectangular to barrel-shaped look to it. You'll see the big note caution here. It is extremely infectious. So we cannot just open this up. You have to usually use a sealed culture. It has to be under at least a level 2 hood or higher. Level 3 would be better because it is pretty infectious. So there's the yeast stage. I'm not going to test you on that because it's not really that unique. What I want you to know is this mold stage here. And you can see it's pointing out the rectangle or barrel shape look. Now, a lot of people get coccidioides mixed up with geotrichum because geotrichum also had a rectangular look, but geotrichum was much more chained together. Chained together, whereas coccidioides is going to have spaces in between. They're not back to back chained, and they're not as large. Geotrichum had larger looking rectangles than coccidioides does. So please be able to separate both of them. Here it is again. This is again the non-stained form. Staining makes everything look so much better. All right, histoplasma capsulatum causes histoplasmosis. It basically involves lung, and they'll go throughout the body. North America river basins, Ohio, Mississippi, Missouri. All right, again, both stages. The stage I want you to know for histoplasma is the mold stage. And it has what we call a round tuberculated macroconidia. Tuberculated just kind of means rough. So there it is in tissue. You don't have to worry about that. Here is the look you need to know. So there is the conidia, and you can see that they look rough. I call them kind of spiky. A lot of times people think this looks like burrs, like cockleburs, if you ever get those on your animals or yourself if you're in the woods. But yeah, that tuberculated macroconidia is the key to histoplasma mold stage. And there it is again, not stained. It's harder to see that roughness, but it's there. And then finally, Paracoccidioides brasiliensis causes paracoccidioides mycosis. Central and South America more here. That's the name Brasiliensis. In this case, you have to know the yeast phase. It makes what we call a mariner's wheel. So if you think of the pirate ships in the movies where they have the spokes that are coming out of the wheel, and that's what they use to guide the ship, that's a mariner's wheel. So here, this is a closer-up view. It kind of has the yeast are doing spokes off of there, looking like a wheel. It's not a perfect representation. Use a little imagination. I always think of it like, oh, I'm sailing to Brazil. That's how I remember this is the mariner's wheel one. All right, and this is the mold phase. You don't have to know the mold phase, just the yeast phase on this one. All right, this is just kind of a quick overview on where you'd find these if you're going to try to culture it and identify it. Obviously, respiratory lungs, best way to find them all. Now, getting into the yeast, a lot of what we've talked about has been molds. That last group was molds and yeast. Let's go into straight just the yeast. Yeast are going to reproduce by what we call budding, what is basically making blastoconidia formation. So they will eventually separate off into a mother and daughter cell. Budding is the best way you have yeast present. You can easily identify, oh, that's a yeast because it's budding. So there it's showing the budding right there for you. All right, here are the clinically significant yeast: Candida, Cryptococcus, Trichosporin, and Malassezia. So when we identify yeast, anytime that you're involving a sterile body fluid, so if you get a cerebrospinal fluid, you better identify any yeast there. Blood, urine, none of that should have yeast ever. If you are taking it from a very immunocompromised patient, always should identify and work up the yeast. Anything that's respiratory that might be Cryptococcus neoformans should be identified. All right, so let's start with Candida. Candida albicans, number one yeast that you will ever run into in the lab. You'll see Candida albicans before anything else. Causes thrush, meningitis, pulmonary infection, eye infection, endocarditis. So again, these are going to be grown on agar plates. They look similar to bacteria. They can be creamy, but they can also be very dry looking. I did grow a Candida, or Alice did last session, and they were very dry colonies when we grew them out. There is a test, though, that we call the germ tube test. And this is basically a germ tube is the first stage of hyphae formation. So when yeast are starting, they look at like that budding stage, but as the yeast infection continues, they'll start forming hyphae. And when you get yeast and hyphae present, you know that that yeast infection has been there a while. When they start making hyphae, it's been there for a little while. So the germ tube is the very first stage in which that hyphae is starting to be formed. Candida albicans, huge producer of germ tubes. So we can do a germ tube test to identify its Candida albicans versus any other Candida that might be there. So if there's germ tubes present, we know it's Candida albicans. So here is what it looks like. It's stemming off of that yeast there. Kind of looks like sperm. I know, a lot of people think that, but it's germ tubes. So when you see, you do need to know this picture. And when you see this, you should be able to say that's Candida albicans. Now, the agar to grow yeast on, you can grow them on blood agar, but there is a special agar called Tween 80, just to keep that in mind. Here's a look at the germ tube again. There are things called pseudo germ tube. I'm not going to test you on this. It's here for your information, but we need the germ tube more than anything. So for the Candida albicans, I'm not going to worry about the pseudo for now. Cryptococcus, oh, you definitely need to know Cryptococcus. If you've taken immunology, maybe you learned about this one there. I know I used to teach it in immunology, but Cryptococcus is found in pigeons, also nature in general. It's hugely associated with AIDS patients. It's very much seen there. Causes meningitis. That's why it's big to identify it out of cerebrospinal fluid. Also pneumonia, septicemia. So you can tell it's a very significant infection when they get it. So Cryptococcus, one of the best ways to identify if we think we have it is to do what we call an India ink stain. And what an India ink stain does is it stains the entire sample basically like this brown color, except Cryptococcus neoformans. Because Cryptococcus neoformans has a very large capsule, and the stain cannot penetrate or get into that capsule. So when they stain the sample, everything will look brown except the neoformans, which will be clear. So it'll look like these clear halos, just like this, because it couldn't get through that capsule. So when you see this, it's a quick way to say, oh, yeah, we have Cryptococcus neoformans here. That's a positive. There are otherwise like kits, latex agglutination kits that you can do as well. And so there's quick identification to let the doctor know, yeah, that patient has Cryptococcus neoformans. All right, overall, here is a way you could work up yeast in a lab. You could do the germ tube. If it's positive, again, it's Candida albicans. If it's negative, you can look at growing out on Tween 80 cornmeal agar to try to see any of the different types of yeast that might be there. Again, Cryptococcus, if you suspect it, you would do like that India ink stain, or if you have a latex agglutination kit. All right, so one mention on this at the end here, pneumocystis. It's so bizarre for me to teach this in mycology because when I learned about pneumocystis, I learned it as a parasite. And so it's so weird to me now to think of it as a fungus. So they've reclassified this into the fungal category. Pneumocystis crinii, Pneumocystis girovetii are the two big ones. Pneumocystis crinii, I've always associated again as a big one with AIDS patients, commonly found with rats, and then girovetii is found in humans. But it's just a note. I'm not going to test you on pneumocystis. It's just an FYI. It is now a fungal. In case you ever did know it as a parasite, you might not have if you've never heard of it. But in your future, just keep it in mind that it is definitely one that does involve AIDS patients as well. There are different stages to it. And then again, because it's a fungus, we can still use that chalcofluor white fluorescent stain. Remember, we learned that at the beginning of Micro 1. That will bind to fungal cell walls and highlight, like it will fluoresce that it's present. So you can use that here as well. So there's just different staining processes. I'm not going to test you really otherwise on pneumocystis. All right, again, this is just a nice other flow chart of working up lab, like different specimens for fungal identification, because it's a bit different than bacteria. Fungal are worked up quite a bit different than bacteria are, but just like anything else, you start with it. You can do stains on it. You can grow it on special agar. You can do like a KOH prep, an India ink stain. It just depends what has growing. If it looks like a yeast, you work it up more like a yeast. If it looks like a fungus on the agar plate when it grows, you work it up like a fungus, which involves definitely microscopic identification. There's a lot less testing to do on a fungus than there is with bacteria. That's probably the beautiful part of it. A lot of it involves just the microscope work. All right, as far as media, there's lots of unique media to grow these on. You do not need to remember any of these media, but they're here for your information. Again, you can incubate it at room temp. A lot of these molds love to grow at room temp, and some at in the incubator, especially because the dimorphic have that yeast phase that wants the incubator, 37. A good 4 to 6 weeks here you're going to be keeping these agar plates for. Here's information on those agars. If you really wanted to read it, you can. As far as treatment goes, there are antifungal medicine out there. The main one is amphotericin B. You're not going to have to remember this. This is just FYI. All right. It's a big one. It's a big start to our week 1. It's a lot of information. Replay this. Write down anything that I said. Know this picture. Know this. That's what you have to know. And when we're in lab together, we're going to go through these pictures. We're going to start to try to remember how to identify them because your quiz next week will have pictures on them. And that is the biggest thing I want you to remember out of mycology is how to identify them. If you know anything out of this PowerPoint, it's the pictures that I told you to remember. That's what I definitely need you to remember first. So if you have questions, save them for me. We'll talk on Thursday together in the lab. Otherwise, have a great week until then. Thanks, everybody.